Fertilization is one of the most remarkable and finely coordinated processes in human biology, representing the very beginning of life, and when visualized in a detailed 3D environment, it transforms into an immersive microscopic journey filled with motion, competition, and precision. The process begins with ovulation, where a mature egg cell, or ovum, is released from the ovary and gently guided into the fallopian tube by finger-like projections known as fimbria, creating the stage where fertilization will most likely occur. At the same time, millions of sperm cells begin their journey within the female reproductive tract, each propelled by a whip-like tail that generates forward motion through rhythmic waves, and in a 3D visualization, this movement appears almost like a synchronized underwater dance. It's driven by both mechanical force and chemical guidance. However, this journey is far from easy, as the sperm must navigate a series of challenging environments, including the acidic conditions of the vagina and the narrow passages of the cervix, where only a small percentage of the original population manages to survive and continue forward. As they progress into the uterus and toward the fallopian tube, sperm cells are guided by subtle chemical signals released by the egg, a process known as chemotaxis, which directs them closer to their ultimate destination. In a highly detailed 3D representation, the interior of the fallopian tube reveals a dynamic landscape filled with fluid currents and ciliary motion, both of which play a critical role in assisting or hindering the movement of sperm. Eventually, a small group of sperm reaches the egg, which is surrounded by protective layers known as the corona radiata and the zona pellucida, forming a barrier that only the most capable sperm can penetrate. At this stage, the sperm undergo a vital transformation called the acrosome reaction, during which enzymes stored in the sperm's head are released to break down the outer layers of the egg, allowing access to the inner membrane. This moment is often depicted in 3D with striking clarity, showing the sperm interacting with the egg's surface in a highly specific and selective manner. After intense competition, a single sperm successfully penetrates the zona pellucida and fuses with the egg's plasma membrane, marking the moment of fertilization. Immediately after this fusion, the egg undergoes rapid biochemical changes to prevent additional sperm from entering, a mechanism known as the block to polyspermy, which ensures that the resulting embryo will have the correct number of chromosomes. Inside the egg, the sperm releases its nucleus, and in a visually captivating 3D sequence, the genetic material from both the sperm and the egg begins to reorganize and prepare for fusion, forming two pronuclei that gradually move toward each other. As these pronuclei merge, they create a single diploid nucleus known as the zygote, which contains a complete set of genetic instructions that will determine the development of a new individual. This moment of genetic fusion is often illustrated as a glowing convergence of DNA strands, emphasizing the significance of this event as the true beginning of life. Following fertilization, the egg becomes activated, triggering a cascade of intracellular events, including a wave of calcium ions that spreads across the cell, initiating metabolic processes necessary for development. The zygote then begins its first mitotic division, splitting into two identical cells, and this process continues as the embryo travels slowly along the fallopian tube toward the uterus. In a 3D visualization, this stage highlights the transformation from a single cell into a multicellular structure, known as a morula and later a blastocyst, with each division carefully regulated and precisely timed. The journey through the fallopian tube is supported by the coordinated movement of cilia lining the tube's walls, which gently propel the developing embryo forward, while also providing a nourishing environment. As the embryo reaches the uterus, it prepares for implantation by shedding its outer protective layer and interacting with the uterine lining, a process that is critical for establishing a successful pregnancy. The 3D perspective allows us to observe how the embryo attaches itself to the endometrium and begins to form connections that will eventually develop into the placenta, ensuring a steady supply of nutrients and oxygen. Throughout this entire process, the interplay between cellular structures, biochemical signals, and mechanical forces demonstrates an extraordinary level of coordination and efficiency, highlighting the complexity of human reproduction. What makes 3D visualization particularly powerful is its ability to reveal details that are otherwise invisible to the naked eye. but allowing us to witness the intricate interactions between sperm and egg, the subtle changes within the fertilized cell, and the early stages of embryonic development in a way that is both educational and visually compelling. From the initial release of the egg to the formation of a fully functional zygote and its journey toward implantation, fertilization represents a seamless integration of biological systems working together to create new life. This process, though microscopic in scale, carries profound significance, as it marks the starting point of human development and the transmission of genetic information from one generation to the next. Through advanced 3D modeling and animation, we gain a deeper appreciation for the elegance and precision of this natural phenomenon, transforming a complex scientific concept into an engaging and understandable visual experience that continues to inspire curiosity and wonder about the origins of life.